Okay, my computer is really slow because the computer is making the program for the Zoom meeting. And once it's done, it'll speed up once it's done. But until that finishes, it'll probably take 10 more minutes. My computer will be really slow. And that's the program I then upload to YouTube. That reminds me, and that is somebody emailed me a question about the lab, and they sent it to my clark.edu email, not the Canvas email. And I'm just warning you that if you send me an email to clark.edu, I will not respond right away because at most I'll be checking that email twice a week. I don't check it every day. And with the Canvas email, I promise I'll check it at least every other day. And frequently I'll check it every day. Any question about any of that? All right. Let me share my screen. Well, if I can get there. Like I said, my computer is slow, so let's go to share screen. Should be able to see my screen, and I don't know if I can get rid of that. If I click on that, there it goes. So today is July 19th, and we're going to be talking about Lab 4 stains. You should read the unknown project instructions, which you can find as a file on Canvas, and you should have it read by next week. I'm going to be discussing the unknown project, and you're going to be starting it next week. Any question about any of that? Can somebody confirm that you're hearing me, please?
Yes, we can hear you.
Okay, great. Let me mention that you're supposed to be working on the infectious disease research project, and you need to get the project approved by me. And most of you have not told me what you want to do. And those of you who have told me, I think both of you, I told you you had to narrow it down or something, meaning somebody stated something like pneumonia, and I said, no, you have to choose one disease that causes pneumonia because there's many different diseases and bacteria species, and there's also fungal and viral species that can cause pneumonia, and you have to choose one. And the same with the UTI, urinary tract infection. You have to have a UTI that's caused by a single species before you do your project. Meaning I'm not going to let you do a project that's too big for the assignment. So you need to narrow the project down. And that's why you need to get it approved by me. Any question about any of that? All right. If not, I'm going to close this down because the more things I have open, the slower my computer will be. And for the next 10 minutes or so, it'll be slow. All right, so there is some reading for this lab, lab 4, staining techniques. We're going to talk about simple stain, the differential stain, and special stain. There's some reading in the textbook about the bacteria size, shape, and arrangement, bacteria cell walls. There's some reading on the simple stain, and that's actually just a page. There's some reading on the differential stain, and that's only about 2 pages. The Gram stain and then the acid-fast stain. And then we're going to talk about special stains, and there's only 2 pages to read about that. Negative staining for capsules, and then endospore stain, and then the flagella stain. There's some video clips you need to read or view for the lab. And I think there's a virtual lab for today. I think so. All right, the learning objectives for today's lab: be able to prepare a bacterial smear for staining the cells. Remember that making the bacterial smear is not staining the cells, and staining the cells is not making the bacterial smear. You need to make the bacterial smear generally before you stain the cells. Understand the principles and procedures used when performing a simple stain, differential stain, and special staining techniques. Be able to interpret the results for a simple stain, a differential stain, and special stains. Be able to define the following terms: what's a simple stain, a differential stain, a special stain, what's a coccus, a bacillus, beryllium, peptidoglycan, gram-positive cell, a gram-negative cell, what's an acid-fast stain, what is the primary stain, what's a mordant, what's a decolorizing agent, what's a counter stain, what is a capsule, what is an endospore, and what is a flagella. Because of their extremely small size, bacteria are nearly transparent when we look at them under the microscope. Therefore, it is necessary to stain them before observing them. And before you stain them, you need to prepare a bacterial smear where you add a drop of water on a slide, and then you add the bacteria to it, and you only add a little bit of bacteria, very low. And then you smear that drop of water containing the bacterial mixture over the surface of the slide. Ideally, you get the bacterial smear only one single layer thick. And then you air dry the bacterial smear and then gently heat it to heat fix the cells on the slide. The simple stain is one that generally uses one dye to stain the cells. And it's usually one basic dye, but it could be one acidic dye. A differential stain usually uses two or more dyes that provides additional information than a simple stain can provide. However, a differential stain does not have to use two dyes. It may only use one. The main differential stain that we'll talk about is the Gram stain. And then lastly, there's another group of stains that we call special stains. And they're for viewing special structures within cells. When we're talking about bacteria, you should realize that bacteria take on common cellular morphologies. First, let me back up and state that bacteria generally can be three different shapes: bacilli, cocci, and spirilla. Do not call them spirilla bacilli. I'm not sure why this slide calls it that, because bacilli are different from bacilli. Spirilla are different from bacilli, so just call them spirilla. Okay? Those are the three main shapes that bacteria have. They also can have cellular growth arrangements. They can have singlets, where you have a single cell. A diplo, like diplococci, where they are in two cells. They can be streptococci, where they're in strings of cells. And they can be tetrads. In higher order, Sarcsinia is 8 cells. And then there are staphylococci. Okay, bacilli have fewer growth arrangements. You can have singlets. You can have diplobacilli, which isn't shown. You can have streptobacilli. And that's about it for bacilli. Okay, Spirilla or only Spirilla, meaning they're all singlets. All right, I've already talked about the Gram stain. It was invented in 1884 by Hans Christian Gram. So you should always capitalize the G when you say the Gram stain because it's named after Dr. Gram. He used it as a differential stain, and then microbiologists started using it, and they discovered that it's differential and separates all bacteria into two large groups, the gram-positive cells and the gram-negative cells. And you know, I didn't mention the archaebacteria also have gram-positive and gram-negative. Of course, I haven't talked about the cell wall, so I'll talk about that when I talk about the cell wall. All right, the Gram stain is the most common differential stain. It uses crystal violet as the primary dye, which stains all cells purple. The cells that retain the crystal violet after the decolorization step are called gram-positive, and they continue to be purple, while the bacteria that do not retain the crystal violet lose the crystal violet, and those we call gram-negative, and they will stain pink after we stain them with safranin. If you didn't notice, the Zoom program just finished, and I saved it down. So my computer will now work a little faster, work better. Here's a typical Gram stain. Let me blow this up, where you can see gram-positive bacilli, gram-positive cocci. There's another cocci. Gram-negative spirals, right there and there. The gram-negative bacillus. I'm not seeing gram-negative cocci here, but there might be some in there. Any question about any of this? The Gram reactions, as I mentioned in the last lecture, are based on the ability of the bacterial cell wall to retain the crystal violet or the crystal violet iodine complex. Gram-positive microorganisms contain a thick peptidoglycan layer in their cell wall, which allows them to retain the crystal violet iodine complex, while gram-negative bacteria have a thinner peptidoglycan layer, and they do not retain the crystal violet iodine complex. So here we're looking at the peptidoglycan layer of a typical gram-positive cell. Let me blow that up as big as I can. You can see the layer is many layers thick, layer upon layer. And here is the gram-negative cell. Move this over. It does have a peptidoglycan layer, and it's many layers thick, but it's much thinner than the gram-positive cell. And the gram-negative cells also have a lipid outer layer of the cell wall. We call this the outer membrane of the cell wall. And then this is not part of the cell wall, that's the plasma membrane. Any question about any of that? some gram-positive bacteria lose their ability to retain the crystal violet dye and are easily decolorized. Therefore, they are described as gram variable. So as you notice, they are classified as gram-positive, but they can easily lose the crystal violet, or they don't take it up in the first place. And so we call them gram variable. Here we have the gram variable cells, and we did the gram stain. This is one species, and as you can see, there are gram negative and gram positive cells here. Let me blow this up as much as I can. These usually happen with the Mycobacterium, and what happens is they have mycolic acid in their cell wall, which is a lipid, and it does not allow the dye of the gram, the crystal violet, to come into the cell and stain the cells. And so they stain poorly, if at all, with the gram stain. They are classified as gram positive because if you do this gram stain well, they usually stain gram positive, almost always stain gram positive. But if you have old cells, they will stain gram variable. And here in this cell, this stain of the gram stain, you can notice some differences. What do you notice about this cell here and that cell here, besides that this cell is pink and that cell is purple? And we can look at it with this cell here and that cell there, or that cell there. What do you notice about the difference between these cells? Come on, somebody's got to notice something. Don't be shy, just take a guess.
They have different shapes.
Even if you're wrong, so what? I'm asking you, what do you notice is different between this cell, that cell, and this pink cell, and that pink cell right here, and this pink cell here? It's very obvious in this cell,
They're different shapes.
compared to that cell and this cell.
They're different shapes.
Well, no, that's still a bacillus, and that's still a bacillus, but there are some differences.
They're different shades of like the stain. Some are darker, some are lighter.
Louder, I'm not hearing you.
They're different shades, so like some are lighter, some are darker.
Yeah, they're different shades. When you look at this cell, it looks a little bit ghostly, especially right here. And the same with that one. And then this one really ghostly, except for that portion right there. Whereas these are distinct. These have indistinct borders. And the borders are very succinct and distinct. What's happening is that these cells are newly growing cells and they're live. These cells are probably dead, and that's why they're not distinct, why their shape is deteriorating. The cell wall is probably broken, like right there, it looks like the cell wall is broken. Okay. And so the dead cells are staining gram negative, and the live cells are staining gram positive. That you'll often find on the Mycobacterium when you're staining old cells. And this is one reason when you do the gram stain that you should only stain freshly grown cells. If the cells are greater than more than 2 days old, you'll have dead cells, and then they may not stain correctly. Any question about any of that? It's particularly seen with the Mycobacterium, but other cells can stain incorrectly if you are staining old cells. The only time I've ever had a student show me a slide like this, they were staining Mycobacterium and the cells were greater than 2 weeks old. And I told them, you can't do the gram stain on cells that are 2 weeks old. It needs to be on freshly grown cells. All right. Some tips to prevent gram variable staining. Avoid using old cells. Always make thin smears. A thick smear will have problems getting the dye into the lower layers. And if you're washing out the crystal violet iodine complex, you'll have trouble washing it out of the lower layers. So you want a thin layer when you make a thin layer of cells when you make the bacterial smear. You should heat fix the bacteria only after the slide has air dried. If the cells have not air dried, you'll get the water to boil when you heat fix, and that'll put holes in the bacteria cell wall. And then avoid overheating the cells during heat fixation, because that'll disrupt the cell wall. And then, of course, do not over decolorize or under decolorize. And that's really tricky. You need to get some practice to understand how to do that. All right. Any questions about the gram stain? If not, let me move on to the acid-fast stain, another differential stain. Bacteria such as Mycobacterium and Nocardia have a waxy substance called mycolic acid in their cell walls. The Mycobacterium have a lot of it. Nocardia just has some. These bacteria are able to resist acid alcohol decolorization, meaning the acid-fast stain does not wash out of the Mycobacterium. For something like E. coli, which is acid-fast negative, the acid-fast stain does wash out of E. coli. When we look at the acid-fast stain, the acid-fast bacteria will stain pinkish, and the acid-fast negative bacteria will either stain bluish or greenish, depending on what the counter stain is. Any question about any of that? So the acid-fast stain, when you use it, it stains the acid-fast bacteria, acid-fast positive bacteria, and it does not wash out of these cells, but it will wash out of the acid-fast negative bacteria. All right, let's move on to the special stains. And the special stain is a stain that is used specifically to stain a special characteristic or property of cells. And it depends on what the stain is, what it's going to stain. Like we have a negative stain and its purpose, or the capsule stain, both of those terms mean the same thing. Its purpose is to stain the glycocalyx, which is something that is firmly attached to the cell wall, and it's external to the cell wall. And if you can see it under the light microscope, we call it a capsule. So it's external to the cell wall, it's excreted by this cell. And we have the capsule stain, also called the negative stain. And its purpose is to stain the cells and the background of the cell, but it does not stain the capsule. I should mention that normally when you work with cells and make a bacterial smear and then go through the steps of the Gram stain, that will wash away the capsule. So the Gram stain will not show up the capsule, and then the Gram stain is not designed to show you the capsule, whereas the capsule stain is. The capsule production by cells is important because it can be associated with the organism's virulence, which means it's more pathogenic. So a cell that has a capsule can be pathogenic, such as Streptococcus pneumoniae or Streptococcus pylorus, which causes strep throat. One of the reasons why those species are so pathogenic is because they produce a capsule, and then the host, their immune system, is not able to reject the pathogen and fight it off. And then strep throat, if you don't know, is very hard for the host to throw off that disease. And if you get it, you usually have the strep throat just get worse and worse and worse. And then it goes on to a secondary infection. When we look at cells that have a capsule, it gives them a mucoid appearance. When we look at the colonies growing on agar, and that's just the capsule, makes the cells mucoid looking. When we look at the capsule with a capsule stain, this says it's India ink, but I know it is not, because India ink stains the background very darkly and the cells very lightly. So this is some other capsule stain, and it is staining the background and it is staining the cells, but it is not staining the capsule. And this is Klebsiella pneumoniae, which is a dangerous bacteria to get because it has a capsule, and then the body's immune system has trouble throwing it off, and it will grow in your lungs and cause pneumonia. And in the old days, you couldn't fight this disease off, Klebsiella pneumonia, and then the patient just got worse and worse pneumonia and then eventually died. Nowadays, we can fight it off with antibiotics. Another special stain is the endospore staining. Endospores are metabolically inactive cells, and the cells make them whenever the environment becomes unfavorable to the cell, and then the endospore can survive, whereas the parent cell will die in the unfavorable environment. So the endospores are hardy structures, and they survive most treatments. It's very difficult to kill endospores. And then when the endospore finds the environment has changed, and now the environment is favorable for the bacteria to grow, the endospore will germinate, giving rise to the vegetative bacteria, which is metabolically active and can grow and metabolize and then reproduce. Any question about any of that? So the important thing to note is that endospores are not a reproductive structure. I don't have it shown here, but one cell will make one endospore, and as the environment deteriorates, the parent cell will die, leaving the endospore. So one cell makes one cell. That's a survival strategy, not a reproductive strategy. So it's not reproduction, but it is survival. This table lists some important endospore forming bacteria, actually gram-positive bacteria, bacilli. And then the diseases they cause, like Bacillus anthracis, causes anthrax. You probably haven't heard too much about Bacillus cereus, but it can cause food poisoning. Clostridium botulinum, I'm sure you heard about, causes botulism, which is a form of food poisoning. Clostridium tetani causes tetanus. And then Clostridium perfringens and Clostridium difficile causing other diseases. Clostridium difficile is often called C. diff, and it causes colitis. And it's hard to treat because the bacteria form endospores. So when you first treat the disease, C. diff, with bacteria, you only kill off the vegetative cells. The endospores will survive. And so you will need, the disease will come back and you'll need to be watching for it. And then the second time you treat it, because the environment has become favorable for the endospores have germinated. And if you catch it early, when C. diff has come back, the second treatment usually kills off a C. diff. All right, any question about any of that? You will need to use this table in the lab today. So here we're looking at some cells that have been stained by the endospore stain. Let's see if I can move that over a little. You'll note that the endospores stain green, and then the vegetative bacteria, which make the endospore, stain pink. Draw this up a little bit. Here you can clearly see the endospore inside the parent cell, and the parent cell here is pink, and you'll notice that the endospore is in, not exactly the middle, but it's inside the parent cell, and it's off to the left, but it's not at the end of the parent cell. This one is close to the middle, but most of them, the endospore is forming off to the left, but not the leftmost region of the cell. That's an endospore that has come out of the parent cell, meaning it's no longer attached to its parent cell. Any question about that? You'll note that the endospore is usually smaller. It's almost always smaller than the parent cell. Another special stain is the flagella stain. It stains flagella. They're long thread-like appendages attached to the cell's surface, and their purpose is to provide the cell with motility. If the cell can swim through liquid media, then it has flagella, or at least one flagellum. You don't need to know this. I'm not going to ever quiz you on this, but you need to look at this part of the lab manual and note that peritrochus is what we call the cell when the flagella are essentially all over the cell and facing every direction. Amphitrochus is when the flagellum are on two sides of the cell, but they're all going, I don't know, east and west. They're all going one way or the other, meaning polar. Lophitrochus are when the cells pass all of the flagella on one side. They're all going one way. And then monotrochus is when the cell has only one flagellum. Okay. Whenever you see a picture of flagellum, you'll usually see more than one flagella type. And that's because, let me see if I can blow this up. This is all one species. This cell looks like it may be putting out one flagellum. This cell looks like it's got flagellum only on one side. That cell's a better example. It only has one flagellum. I'm not seeing flagella on both sides, but that's not important. Maybe that one has flagellum on both sides. And then this one is peritrochus, that one is peritrochus, and this one is peritrochus. The reason why these cells have less than the peritrochus flagellum is that they're just making the flagellum. This one's only made one flagellum, but given more time, it'll put out more than one. And given lots of time, it'll probably become peritrochus. Okay, so when you're discussing the flagellar arrangement, you put up the highest number you see. Meaning on this slide, you'd say it is peritrichous because that's the highest arrangement. Okay. And the other ones, like the monotrichous, it's just getting started making the flagella. And presumably with more time, this cell will eventually become peritrichous. All right. So you do need to know this for answering one question in the lab, which is why I'm discussing it. But you do not need to know it for the quiz. So you don't need to memorize it. You should know the terms of the lab. You can skip the references. One student once went looking at the references, and one of them was a link to an infected website, which, what do they call that? It had a worm on it or a bug on it, and then that infected the student's computer. And we apologize for that. Nobody had ever checked the references before. And it wasn't our fault. We got this from somebody else, and they got it from a site that was infected. So it's a good thing sometimes not to check the references, but as far as we know, all the references are good. All right, laboratory 4 exercises. You're not physically going to be performing any of these activities, but you must understand how to prepare bacterial smears for staining and how to make the bacterial smear. On procedure 1, you're going to be preparing a bacterial smear for staining. You're not going to be doing the stain, you're just going to be preparing the bacterial smear. So watch a video on preparing a bacterial smear, and then watch a video on heat fixing the bacterial smear. And then read through the making the bacterial smear. Then in procedure 2, we're going to be looking at a simple stain. So there's a video clip for watching a simple stain. So watch that, and then review the steps for doing a simple stain. Essentially, you're adding one dye. It can be either a basic dye or a negative dye. And you put it on the cells and then wash it off. And then all the cells will stain the same. So they're all purple, the E. coli and the Staphylococcus aureus. So you can't notice a difference in the color between the cells. And then I think we have a question here. Describe the cellular morphology of E. coli. And I'll warn you that in some places, like right here, and maybe right there, right there, maybe right there. It looks like that they're not bacilli. It looks beryllium or spirals or bent rods. And that's because this is one rod there and another rod there. This is one rod here. And this is probably 3 rods, one there and then one there. And you can't really see where the different rods are in this picture. Okay. So the correct answer for E. coli is they are a rod or a bacilli. And then give the total magnification. And then move on to procedure 3, the gram stain. We have a video clip on performing the gram stain, and we have a virtual lab. But probably not going to open. So let me see if I can grab it this way. Come on, there we go. Once you go to this website, it will take time to download onto your system, particularly if you have a slow internet, but it should download in just a couple of seconds. That's weird. Oh, I know what the problem is. This is probably the old procedure. Let me see if I have the new one up. Now this might work. This is Microsoft Word. Nope, just taking a long time. Yeah, this works. All right, so click continue to get it to run.
Hi, Lab Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria. All yogurt has bacteria in it since yogurt. Unfortunately, this is what plain, good yogurt looks like. Notice the uniform texture and color. Now take a look at this yogurt. See how the whey and water are separating? Are those air bubbles forming near the bottom of the bottle? These may be signs of contamination.
You got a little video there, which it's interactive, and I don't remember if there's something for you to do in it or not, but for the gram stain, excuse me, I took that off because it was so loud. I remember why it's so loud. That is, I was having trouble hearing the students, so I turned up the volume. Let me turn the volume down. All right, so for doing the gram stain, click this here, and then get started with the gram stain. So you're actually going to be doing the gram stain in a virtual
Hi, Lab
lab.
Assistant. Welcome to the yogurt section of the dairy processing plant. It appears that our latest batch of yogurt might have been contaminated by some undesirable bacteria.
This looks like it's the same thing that we were looking for.
It's
still too loud for me. Let's try it there.
Before you start, there are basic safety procedures that need to be followed in all labs. Wash your hands before and after handling the materials. Wear gloves and safety glasses. Don't eat or drink in the lab. Be aware of dangling jewelry, loose clothing, or long hair that might get caught in equipment. Clean equipment after use to avoid the possibility of contaminating the next person who uses it. Always strictly follow the instructions given. All yogurt has bacteria in it since yogurt is made by adding specific bacteria to milk. This is what plain good lung tissue.
All right.
Why do bacteria stain pink or purple?
So it does go through the
Bacteria that are
gram
in the
stain
yogurt.
for you.
Then we'll look at them under the microscope. The bacteria will be stained different colors depending on what type they are. Gram staining. Hans Christian Gram devised this technique in the 1880s to make bacteria more visible in stained sections of lung tissue. Today, the technique is used as a tool to differentiate gram-positive and gram-negative bacteria as a first step to determining the identity of a bacterial sample. Bacteria that stain a pink or reddish color like this are called gram-negative, and their presence means the yogurt is contaminated. Bacteria that are used to
Okay, go through the video. I'm looking to see if it goes through the gram stain. Yes, it does. So right
small
here,
sample of yogurt will be placed. Turn
We're
the
starting
slide
with gram
over
stain.
so the markings are on its underside. Click and drag on the slide to draw a circle in the center of the slide. As in the previous scene, click and drag on the slide to label it with the name of the sample source, for example, yogurt. This will prevent confusion between this slide and others that you may be studying in the lab. We need to dilute the yogurt before we put it on the microscope slide. We'll use a PBS liquid, phosphate buffered saline, as the diluent to thin out the yogurt. That helps us see the bacteria with the microscope. Sample, which is important because all organisms exist in a very narrow to balance pH. Start by placing 1 milliliter of the diluent into a test tube using a pipette. What is a pipette? A pipette dispenses a measured volume of liquid. This picture shows a transfer. We'll use it to place a small amount of yogurt into the test tube with the diluent. Start by sterilizing the inoculation loop in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot. Work close to the Bunsen burner. Remember to always work close to the Bunsen burner, about 1 foot away. This will prevent contamination of your sample by microorganisms in the air. After it has cooled down, use the loop to obtain a small sample of yogurt from the suspect container. To get a good sample, reach deep into the container or thoroughly mix the yogurt first. Place the yogurt sample into the previously measured diluent, PBS. Now, gently shake the test tube for 5 to 10 seconds to thoroughly mix the yogurt sample throughout the diluent. Use the inoculation loop to transfer a small amount of sample from the test tube to your prepared slide. Sterilize the inoculating loop again in the flame of the Bunsen burner. Hold it directly in the flame until it glows red hot. Transfer a small amount of sample from the test tube to our prepared slide. Place 2 loops full from the sample onto the slide. Tilt the slide to spread the drop out slightly, about the size of a penny. Now we'll heat fix the slide. This prevents the bacteria from being washed away when we begin the staining process.
They should
Place
air
the slide
dry.
in a
I didn't
clasp,
mention that.
such as a clothespin, and pass it over the flame of the Bunsen burner. Be careful not to put it directly in the flame. We don't want to burn the sample. Just dry it out so bacteria won't wash off. What happens if the sample is overheated? The proteins in the cells denature, causing the bacterial positive bacteria. What is crystal violet? It is
All
a
right.
violet-colored dye.
All right, so from here now, it's going to start with the gram stain, which is the main reason why we got this, although it does help going through making a bacterial smear. Any questions about any of this? All right. So do the gram stain under the virtual lab. And then review the steps for making a gram stain. Review what cells are stained at what steps in the gram stain. You do need to know this, by the way. And the lab will ask you, what is the color of a gram-negative cell after you apply the mordant? What is the color of a gram-negative cell after you apply the decolorizing agent, etc. And then answer the questions in the lab. So it's going to ask you, what is the gram reaction for E. coli, gram positive or gram negative, and what's the cellular morphology, the shape, is it coccus or bacillus? And then for Staphylococcus aureus. And here we'll see a difference in the color, which we can here, Staphylococcus aureus and E. coli, because it's a differential stain. All right, so for each of the following organisms, answer the question, predict the color you'd see if the color of the cells are pink, purple, or colorless after each step in the gram stain. Then do procedure 4 for acid-fast bacteria. We have a little video clip on doing an acid-fast bacteria stain. We're going to be performing the modified Kenyon acid-fast bacteria stain. And there's the steps here. I'm not going to read through it. You can read through it. The important thing is that we have a question here, and sometimes students leave this. The question for procedure 4 is right here. It doesn't have a question mark. Interpret whether the organisms denoted by ARIS or AFB, meaning acid-fast bacteria positive, or acid-fast negative bacteria. So this is a question. You need to state whether these are acid-fast bacteria, this arrow, whether these are acid-fast bacteria positive or negative, and the same here and for here. So there's 4 questions to ask, answer, and you can put in the answer here and here or here, or you could just write it in leftmost arrow is whatever, second leftmost arrow is, and then state what it is, and then the third leftmost arrow, state what it is, acid-fast positive, acid-fast negative, and then the 4th leftmost cell here, or arrow is. So answer that, and then move on to procedure 5. This is special stains. We have a video clip on the endospore stain. Watch that. We have a video clip on the negative stain. Watch that. And then we have 3 slides here. This is for the endospore stain. This is actually the flagella stain, and that is the negative stain. And then answer the questions for each of the 3 slides. So A applies to slide A. It tells you this is the Schaefer-Fulton stain, which uses what stain as the primary stain, and ultimately dyes what structure that are produced by some bacteria to aid in survival. And then list some examples of pathogenic endospore producing bacteria. Some is more than 1 species. So if you only list 1, you'll get that wrong. Okay? B, that applies to slide B. This is what stain that coats long whip-like appendages which are used to aid in motility. And then state the flagella arrangement seen in the slide. And then 3, list some examples of pathogenic endospore producing, excuse me, I'm on the wrong place. List some examples of clinically significant bacteria that produce flagella. C applies to slide C. And it says this is what stain, so name the stain, that uses India ink or nigrosin. It can be used to detect the presence of what structure in a cell. The structure is located exterior to the cell wall and can prevent phagocytosis. All right, so answer those questions and then list some pathogenic microorganisms that produce capsules. And you can find a list in the table in the lab module. Let me remind you that when you're doing the lab module, it's probably best to read the lab module, but I do not want you to submit the lab module. So do not put your answers in the lab module. Put your answers in the worksheet. The reason is that the worksheet is smaller and it'll take up less room on my Canvas website. And you will hate it if my Canvas website overfills because you won't be able to submit your assignments. And I would have to get more space and start deleting things. And I would prefer not to do anything with Canvas. So don't overfill my Canvas website. If you do, I may apply a penalty. So only submit the worksheet. All right, when you're done with that portion of the lab, then do the 2 case studies. Case study 1 is a 45-year-old female experiencing respiratory issues, meaning she has an infection in her respiratory system. And then based on her clinical presentation, the doctor orders a chest X-ray, which you can see here, and they do a sputum, meaning she spit on a slide, and then they did an AFB culture and stain. AFB is acid fast stain. And then we're looking at the Ziehl-Neelsen stain here. Based on the findings below, answer what is the probable diagnosis. And you must name the species causing the infection. So don't tell me, oh, well, she has a respiratory disease. You have to tell me what species is causing this infection. And you should be able to get it from the case study and the information given here. For case study 2, we have a 64-year-old diabetic patient, and he's admitted to the hospital with soft tissue necrosis on his foot. A routine culture and gram stain are ordered. The bacteria grew anaerobically, and they do not grow aerobically. Based on the findings of the gram stain and the Schaefer-Fulton stain, list the species which is causing this infection. And you can look in the lab module in the table and come down with the species that is probably causing this infection. So to get a correct answer, you have to list the bacteria species. Okay, any questions? If not, work on the lab. I will be here until 9 o'clock or until the last student logs off. So far, all students have logged off much earlier than 9 o'clock. When the last student logs off, I too will log off. Any questions? Okay, if not, go ahead and start working on the lab. And remember,